Hi, everybody. My name's Kelly Pratt, and I would like to present the infamous Dr. Subbiah. This session is called Designing Clinical Trials to Demonstrate the Clinical Benefit in Precision Oncology. Dr. Subbiah is a clinical trialist and physician investigator at the University of Texas, MD Anderson in Houston. He is an associate professor in the Investigational Cancer Therapeutics Department. He's the Center Clinical Medical Director of the Clinical Center for Targeted Therapy Cancer Medicine Division. Dr. Subbiah has a unique training background, as he is one of the few physicians in the nation or the world to have training in oncology in both adults and pediatrics. He's board certified in both internal medicine and pediatrics, and has completed fellowships in both adult and pediatric oncology at MD Anderson. He is primarily involved in translational cancer research and the and conduct of early phase biomarker-driven clinical trials, with a special interest in antibody-drug conjugates, radiopharmaceuticals, immunoconjugates, and basket trials. And that was a mouthful. So Dr. Subbiah, if you wrote this and made me say all of that, I want to thank you.
Thank you, Kelly, for the generous introduction. I think that's it's been, more than all this. What you've read out is probably from when I got recruited like 10 years ago. Right now, mostly, it's been a pleasure and honor to work on RET inhibitors and selective RET, and BRAF inhibitors. And I've had, have the pleasure and honor to take care of, hundreds of patients with thyroid cancer here, together with our amazing, phenomenal endocrine oncologists here at MD Anderson and endocrinology group here. And it's been a rewarding experience to, offer these targeted precision medicines and see the clinical benefit in real time. So, without any further discussion, thank you, Kelly, and thank you, Gary, for this opportunity for me to talk. So let me talk about designing clinical trials to demonstrate clinical benefit in precision oncology. And how we need to be fast, we need to be very agile and plastic in terms of designing clinical trials and moving these drugs to the right patient, to the right clinic at the right time. So the background you see behind my screen is from the Intercontinental Hotel in Paris. I was just there a couple of days ago. It was a presidential symposium, and it's a historic place, where so many historical events have happened. So that is a backdrop you see. So I'm available on Twitter, and Kelly and Gary know how to get hold of me if you guys need to talk to me anytime. So again, what is precision oncology? So we are moving from the era of one treatment for the average patient using a trial and error method that is a traditional practice, to moving to precision oncology, customizing the right treatment for the right patient at the right time. The traditional practice of medicine is non-biomarker driven, whereas the precision oncology paradigm is biomarker driven. We have, in 20 years ago, they said we cannot sequence a human genome, and then they spent $3.3 billion to sequence one human genome. Right now we can get our whole genome sequencing or exome sequencing done for $1,000. So again, we are discovering more and more genes, and as much as we discover genes, we discover targeted therapies to go after those genes. And one of the targets is RET. So let me talk about RET inhibitor, my brief experience about RET inhibitor, a translation of these RET inhibitors from bench to bedside, and how we could move these RET inhibitors within 3 years to a full FDA approval. Brief agenda is RET aberrations in oncology, RET alterations in thyroid cancer, lung cancer, and multiple other cancers as well. And also multi-kinase inhibitors. Of course, we had vandatinib, cabozantinib, lenvatinib, and all other drugs, and why we needed these selective RET inhibitors, selpercatinib and pralsetinib. So RET is a receptor tyrosine kinase required for normal development. and oncogenic RET signaling. So RET gene can be activated by two major mechanisms in cancer. One is the RET mutation, which is seen in medullary thyroid cancer, and RET fusions. RET fusions are rearrangements, which is most commonly seen in lung cancer, 1 to 2% of lung cancer, and 20 to 20% of papillary thyroid cancer, anaplastic thyroid cancer, and differentiated thyroid cancers. So these are the major mechanisms by which RET is driven. Mutation is there is an abnormality in the gene, whereas in the case of fusions, the RET gene itself is okay, but there is an abnormal gene that fuses with the RET gene to make it abnormal. So that is what is fusion. So again, in 1985, RET was one of the first oncogenes to be identified as an oncogene. And in 1990, they discovered that papillary thyroid cancer harbored RET fusions. In medullary thyroid cancer, it was identified in 1993. In 2012, just 10 years ago, it was discovered in lung cancer. In lung cancer, they found that RET fusions were drivers in a subset of lung cancer. Subsequently, we had several cancers like colon cancer, salivary gland cancer that had RET alterations. In 2017, we had in the clinic 2 selective RET inhibitors appeared, Blue 667 and Loxo 292, which is Pralsetinib and Subbiah. And within 3 years, from just entering first in human clinical trials, 2 of these drugs were approved. And I'll talk to you about further drugs that are coming out in the pipeline. So let's talk about RET. As I said, RET is activated by 2 major mechanisms. In non-small cell lung cancer, it is RET fusion. In papillary thyroid cancer, it is RET fusion. In other cancers, it is also RET fusion. In medullary thyroid cancers, and again, as you all know, we see this in over 60% of sporadic medullary thyroid cancer and over 90% of hereditary medullary thyroid cancer. Again, here, the more we test, the more we find. And we are finding more and more RET alterations in medullary thyroid cancer patients beyond the common M918T. Again, we are seeing several activating point mutations that have been described in literature to date. And beyond these, as more and more we sequence, we are able to identify more RET point mutations that activate the pathway. And as you can see here, the most common RET mutations in medullary thyroid cancer are the M918T and the C series, the 634F, G, R, S, W, and Y, and the gatekeeper mutation, the V804M, the L790F, and the Y791F. Fusions. So papillary thyroid cancer and well-differentiated thyroid cancers, 10 to 20% could be driven by RET fusions. The most common pathognomonic fusions in papillary thyroid cancer are this fusion called CCD6, and and the second most common is the NCOA4, right? So why did we need selective RET inhibitors? Again, we had these drugs like cabozantinib, vandetanib. Again, they benefit patients. The challenge with these drugs is they also hit something called the VEGFR, the vascular endothelial growth factor receptor, right? And because of this, right, they see hypertension, hand-foot syndrome, proteinuria, QT prolongation, thrombosis, and hemorrhage. So you can see here, the dose reduction rate, right, is considerably high, and many of the patients could not tolerate these drugs. So this led to discontinuation. So we needed, better drugs to go after RET. And same in the case of, lung cancer, the drugs did not confer a benefit like other drugs for RET fusions. So we needed a selective RET inhibitors for RET fusion positive lung cancer and RET mutant medullary thyroid cancer. So one of the first drugs to be developed was the selpercatinib or Loxo 292. And as you can see, this shows high kinome selectivity for RET. And we showed that in preclinical models, in multiple preclinical models, that this was better than the multikinase inhibitors. This was specific. And most importantly, it covered the gatekeeper alteration, the V804M. The V804M is essentially a mutation that is an acquired resistance mutation, or can be also seen as a germline alteration in patients with medullary thyroid cancer. And none of the drugs like cabozantinib or vandetanib can target that. So this LOXO-292 or selpercatinib was designed to go after these gatekeeper alterations. It also showed blood-brain barrier penetration as well. So we designed this clinical trial and a 3+3 design. And slow and steady, we started enrolling patients from 20 milligram once a day, then 20 milligram twice a day. And slowly we reached to 160 milligrams. So 160 milligram was the recommended phase 2 dose. In fact, currently that is the FDA approved dose, 160 milligram twice daily. And many of the patients who were enrolled at a lower dose level were also subsequently intra-patient escalated to 160 milligram twice daily so that they can all get the benefit. And you can see that why we needed 160 was, we wanted to make sure that we cover the RET M918T and most common RET with this dose level. So I think we will need at least anything above 80 works, but 160 should definitely cover that very deeply. So this trial was designed, and in non-small cell lung cancer, as you can see, these are waterfall plots. These show the shrinkage of the tumor of target lesions. And in non-small cell lung cancer, selpercatinib provided anti-tumor activity regardless of the RET fusion partner. In thyroid cancer, the clinical benefit was observed across multiple thyroid cancer histologic types, and the responses are definitely ongoing. So this was the data. Based on this data, selpercatinib received accelerated FDA approval for RET fusion positive lung cancer and RET driven thyroid cancers that include RET fusion positive papillary thyroid cancer, RET fusion positive all thyroid cancers, and RET mutant medullary thyroid cancer, germline or sporadic RET mutations. The other drug to go after RET was Pralsetinib 667. This was designed to treat RET altered cancers. Again, as you can see, this drug also had high kinome selectivity for RET. And this, as you can see here, was more potent and selective than several of the other multikinase inhibitors. And at that time, the multikinase inhibitor was vandatinib and cabozantinib, and it was shown that Blue 667 was potent against the known ones, and also another experimental RET inhibitor that was being developed at that time. And as you can see here, pralsetinib potently inhibits RET alterations and the gatekeeper mutation while sparing the VEGFR2. So that was important. The VEGFR2 sparing was important to prevent the potential side effects from VEGFR2 inhibition. Again, this trial, known as the ARROW study, was designed. This was a first in human clinical trial. And they started at a low dose of 30 milligrams, and slow and steady, it was increased to 120, it was increased to 190, 200, 300, and now 400. So 400 was the maximum tolerated dose. And once that was reached, once daily dosing, 400 milligram once daily, several expansion arms were designed. So just from a phase 1 study, we identified the dose. And beyond that, instead of going to a phase 2 study, immediately the protocol switched to a phase 2 dose expansion study. So these are efficient basket clinical trial designs that can cut short the timeline of drug development. Because usually if it's a phase 1 study, when you're designing another phase 2 study, the time it takes to establish the protocol, establish the sites, do everything is shortened. I think we need drugs sooner to the clinic. So these kind of phase 1-2 basket study designs help with cutting the timeline of drug development. So early we showed dose-dependent exposure of RET pathway inhibition. So we did pre and post biopsies in patients to show that this drug worked, and we showed downstream inhibition of RET, MERK, ERK, and several other targets like dual phosphatase 6 and SPOUTI4 mRNAs. And this graph here shows the steady state pharmacokinetics in plasma and brain. And you can see here the tumor markers, the carcinoembryonic antigen and the calcitonin also declining based on the dose. So this trial, the ARROW study, as I said, was designed, and the phase 2 expansion is ongoing. The primary endpoints were, again, shrinkage of the target lesions. All right. So again, this was designed similar to the Libretto study. The ARROW study also showed shrinkage in lung tumors. As you can see here, a clear objective response rate of 73%. These are known as waterfall plots. And these show the target organ shrinkage. So if you see the graph below, anything below 30%, right, the cutoff mark, that's called a response, right? But you can see most of the patients definitely had a shrinkage of their target lesions. And same in the case of medullary thyroid cancer. 74% of patients with medullary thyroid cancer, there was a shrinkage of the tumor, more than 30%, right? Almost all patients had some shrinkage of the tumor, with the exception of maybe one patient. And 60% and 74% of patients were treatment naive. That means Pralsetinib was the first drug they received, and 60% of patients who were treated with cabozantinib or vandatinib or both also had a nice tumor shrinkage there. So let's talk about the RET inhibitor timeline. RET identified as an oncogene in 1985. 1993, it was identified in medullary thyroid cancer as a driver. 2012, it was identified in lung cancer. 2017, we had in the clinic, we had the first generation selective RET inhibitors. And I do clinical trials, and I know which drugs work and which drugs don't. So this started working right from the first patient on. We knew as a drug developer that this baby would graduate. It was so rewarding to see dramatic responses in patients with these selective RET inhibitors, both LOXO-292, which is selpercatinib, and BLU-667. And it was rewarding to follow these patients and deriving primary benefit from treatment, and seeing the less number of side effects that we were used to seeing with other multi-kinase inhibitors. In 2020, in the midst of a pandemic, approximately 3 years from when we started first in human studies, right? These two drugs, selpercatinib and Pralsetinib, were FDA approved. Again, these are a power to the testament of precision medicine. Again, usually when you hear drug development timelines, you hear as 10 years, 15 years, 20 years. So this is 3 years from first in human clinical trials. So this is, again, this should not be an outlier, right? I think we have seen in the COVID pandemic that drugs can be developed phase 1, phase 2, phase 3, all within 1 year. The COVID vaccine was developed within like 1 year. The COVID, oral therapy, Paxlovid, was developed within a year as well. So again, this can be done when all hands on deck. Again, for a rare biomarker, 2 drugs, not 1, but 2 drugs were FDA approved approximately 3 years from when they entered clinic. So, what we see right now, I think I see a question here. What is happening with the trial of the drug after ETEMO? So I'm going to start answering that question here. So we are right now in 2022, September, and what we've identified several resistance mechanisms. And unfortunately, that's a case with any tumor is it recapitulates the Darwin's theory of evolution. What happens is what we see is with these drugs, selpercatinib and Pralsetinib, over time, in some patients it works for 2 years, some patients it works for 3 years, some patients do not develop resistance. So the tumor, finds ways to overcome resistance to these drugs. So what we found was there is a mechanism, specific mechanism of resistance called the solvent front alteration, right? What is a solvent front alteration? What happens is if the RET mutation is there and these drugs, selpercatinib and Pralsetinib, can bind and block these RET mutations. So this forms a mutation that prevents the latching of these drugs to this target. So that's how it develops a resistance. That's what we call as on-target resistance. All right. So beyond the on-target resistance, we also see, an off-target resistance mechanism through multiple other pathways. So for on-target selective resistance mechanisms, the good news is that we have in clinical trials, first in human clinical trials, several selective second-generation RET inhibitors that have entered phase 1 studies. And again, I listed a few of those, and I see a lot more others coming as well with different mechanism of action. And the drugs I've listed here are the LOXO-292, beyond LOXO-292, which is the LOXO-260, the HMO6, the TPX0046 and the EP0031. These are the ones that are entering first in human clinical trials. I think in a year, we will hear more updates about these drugs. And they are very early in drug development. And hopefully, we will see how we can, effectively, render clinical benefit with these drugs, selpercatinib and pralsetinib, and if they acquire resistance with these second generation drugs, so that we can transform the lives of our amazing patients with who are motivated, who seek these clinical trials. So in summary, how do we design clinical trials for precision oncology is to deliver the right intervention to the right patient at the right time. The key is establishing a biomarker, right, in the tumor. Here, in this case, RET is the biomarker in the tumor. And, this is a case of an unmet need, huge unmet need. And we didn't have any other drugs that were, so effective in this space. Importantly, we had compelling preclinical studies, and we had strong early phase proof of principle and proof of concept studies. And again, we already knew that there was a high pretest probability of response in our minds when we saw this clinical and preclinical data. And of course, we need to have a reasonably well-tolerated side effect profile. And we also need to see the benefit versus the risk profile in these in this. And here, in RET altered cancers, there was no selective RET drug before, and we had these two drugs. And overall, the benefit outweighed the risk of development of these drugs. Most importantly, as we move further into the future, and what I personally want is more patient engagement in clinical trials, more patient engagement when we design clinical trials. And we want to make sure when designing clinical trials, we want to include patient-reported outcomes, how patients feel, and we want to make these trials from clinical trial centric. So again, most of the trials that have been designed before the pandemic were all clinical trial centric. I would say personally that pandemic has been the major disruptor, and it gives not just investigators but regulators and sponsors of these trials to think about putting patients in the center of drug development, that what we call as patient-centric clinical trials. And I want to see, hopefully we will be in a post-pandemic era soon, and hopefully the lessons learned from the pandemic, we move on completely from clinical trials-centric clinical trials to patient-centric clinical trials, with patients being the centerpiece of drug development. Again, I thank all the motivated patients and their caregivers for their participation in clinical trials, even through the COVID pandemic. And again, from the bottom of my heart, if every patient that enrolled on this clinical trial contributed to the FDA approval so that real patients benefit in the real world, not just in the US, but globally. Right? These drugs are now approved in the EMA and many other countries. I thank all our patients with the bottom of my heart. And thanks to you, all the study investigators, support staff, the sponsors, again, the drug sponsors, no drug equals no trial. So they are making the drugs for us. So we need to be thankful for them. FDA staff. Again, the first drug was approved in May 2020. That was a peak of the pandemic. I think FDA staff worked very hard to deliver these approvals, for these patients, and all the independent data monitoring committees. Again, this is my contact information. I thank, Gary and Kelly and everyone here for inviting me to participate. And I'm also available on Twitter. And thank you so much for the opportunity. I'll be happy to answer any questions. Thank you.
Yes, Dr. Subbiah, thank you. That was very awesome. Could you possibly touch on, before we get to the next questions, the first question where you talked about the second generation, the LOXO 260, HMO, TPX, and EP. Could you give us any snippets of how they're doing, other than, how is anything really catching your eye?
So, several of these studies are just opening. right? They're just starting. They just started because, I would say because of maybe because of the pandemic. These were, these took a long time to open, and now they've started the clinical trials, and hopefully in the near future we will see the clinical data from these studies.
Okay, awesome.
They are still dose escalating, right? They are still slowly dose escalating. So we will see the clinical trial data soon from these studies.
Awesome.
Will there be studies in the near future to find treatment for advanced Hürthle cell metastatic disease? Yes, absolutely. I think what we are seeing here is cancer is a disease of the genes, and we are dividing, cancer into many genomic subsets, right? And what we see is that there is histology specific drug development and a histology agnostic drug development. And what many of these rare cancers, for instance, I would say medullary thyroid cancer and RET hugely benefited from RET being discovered in lung cancer. And 2012, it was discovered in lung cancer, and all the companies and everyone put funding there, and they started developing drugs. And we need to find the linchpin or underpinnings of biology of Hürthle cell cancer and see what we can go after that. Again, we are sequencing all these patients. I think in the near future we will I would say never say never, right? We will find targets for these for targeted therapies for these rare diseases.
Okay, I've got one. Can you speak on second generation inhibitors and clinical trials for anaplastic thyroid cancer? Thank you for all you do. Follow you on Twitter and can feel your excitement with your work.
Thank you so much. That was generous. So again, as you all know, I also developed was part in developing the BRAF MEK inhibitor combination, the dabrafenib trametinib combination for anaplastic thyroid cancer. Again, anaplastic thyroid cancer, as you know, is a challenging disease, and we found that a subset of patients, at least 20 to 40% of patients, harbor the BRAF V600 alteration, and we could target them with dabrafenib and trametinib. So right now we have 3 different agents that are similar to dabrafenib, trametinib, like vemurafenib, cobimetinib, and encorafenib, binimetinib, also that target the BRAF. So beyond these 3 classes of agents, because BRAF V600E is seen in melanoma and many other cancers, again, we have like several second generation inhibitors that are in clinical trials, and several coming in space. There is a BioGene study is there. It's a second generation BRAF drug and in combination with the MEK inhibitor that's available for not just anaplastic but for all patients. Pfizer is developing a brain penetrant BRAF inhibitor that was also in clinical trials. And there are also there is another company that's also coming up with something called a BRAF V600 degrader. Right, that's also entering clinical trials soon, and we are really looking forward to data from these clinical trials that can help anaplastic thyroid cancer patients.
Awesome. There's an anonymous person that wants to say thank you so much for all the work you do for us and others with cancer.
Thank you so much. Thank you.
I've got another one. This is from Ben. I'm a 39-year-old papillary thyroid cancer patient with lymph node metastasis. As of September 8th, 2022, I'm taking larotrectinib 100 milligram twice daily. I'm having some side effects that are borderline debilitating: constipation, fatigue, leg weakness, rare balance issues, brain fog, rare dizziness, mouth and throat numbness, and just a feeling of being off. How long should I expect these side effects will continue at this level, at this current dosage?
Yeah, thank you for that question. I'm so sorry that you are experiencing these side effects. Maybe Ben, you want to? I can talk to you in detail later. You can. My contact information is here. You can reach out to me, and we can discuss offline how we can deal with this. Some of these are unfortunately on-target side effect because TRK. I gather if you're taking larotrectinib, it's an NTRK inhibitor. And NTRK is TRK, tropomyosin-related kinase, is seen in normal cells as well. So when we are hitting the cancer, we are hitting the normal cells as well. Some of these can be mitigated mainly by holding the dose or dose reduction. And we have some drugs that can help with the balance issues as well, such as meclizine. So depending upon which geography you are, I think you probably need to work with your oncologist, and I'll be happy to connect you with our team here to see how we can mitigate some of these side effects. And again, this is a huge quality of life issue, right? Even if the tumor shrinks, you want to make sure that the brain fog, dizziness, and all that, you want to make sure that you want to mitigate that. I'd be happy to talk to you offline about that.
Thank you, Dr. Subbiah. Vilma.
Kelly can definitely connect you with me, and I can share with you what can be done. In fact, if you want to connect your oncologist with me, I'll be happy to talk to them.
Thank you. That's great. And I do have Dr. Subbiah's contact information for everybody if anybody's missed it. Let's see. Vilma wants to just thank Dr. Subbiah for everything he does. I know we can all agree with that. Renee asks, is funding a problem for people like you doing cutting edge research?
I think funding is always a problem. I would say, but we've been successful in working with pharma companies, NIH, to get funding here. One of the main challenges, what I see, is beyond funding. Researchers working together is a problem. Everyone works in silos, right? I think we need more people to work together as a team to answer these questions. Single individuals or institutions cannot definitely answer all these questions. I think there has to be a way where everyone can work together, like academia, pharma, the FDA and NIH together to solve these problems. Again, so far it's been everyone working in silos and individuals pushing the needle. And I think there is a lot more room if we can get all hands on deck here.
Okay, another person, you're popular, says, thank you for all you do to help us in this fight. I've got a question from Susie. Will trials be developed for BRAF and MEK inhibitors for non-ATC papillary cancer? Until there are trials, FDA will not approve and insurance will not pay, even though these are effective therapies. Thank you.
Susan, that's a great question. So that was an issue we addressed recently. So now, as you know, BRAF and MEK inhibitors are approved for all solid tumors in a tissue agnostic manner. So it does not matter what cancer they have, as long as they have the BRAF V600E alteration. The BRAF and MEK inhibitor, specifically the dabrafenib and trametinib combination, has been approved, and it's been working. Just before the pandemic, we started working on that, and it's been a lot of back and forth with the FDA. And recently, on June 26th, it got approved, and so it is approved for non-ATC papillary thyroid cancer as well. As long as any solid tumor patient harbors a BRAF V600 alteration, the drug is approved. So that is the importance about tissue agnostic drug development. So there are many rare diseases. It's going to be very, very hard for institutions, companies, and groups to invest in small diseases. But if it's a disease drug is approved in a tissue agnostic manner, rare diseases benefit. So right now, the dabrafenib and trametinib combination was initially approved for melanoma, then for anaplastic thyroid cancer, then for non-small cell lung cancer. Now, as of June 26, it's approved in the US for all solid tumors that harbor a BRAF V600 alteration. That includes papillary thyroid cancer, HER3 cell cancer, among others. And insurance should approve because it's now FDA approved.
Dr. Subbiah, I've got a good question for you.
Yes.
And you might not want to answer, but we'll see. What are your thoughts on the CAR-T trials through UPenn that are trying to target solid tumors instead of, I know they've had some success with the blood cancers, but what are your thoughts on how that might work out for thyroid cancer?
I think that's a great question. The thing is that with these targeted therapies, we see they work quickly and they acquire resistance mechanisms quickly as well, right? The ultimate goal, if we can arm the immune system to get rid of cancer, right? Any cancer, I think that will ensure that we arm the immune system and we can eliminate cancer. I think that would be golden. The challenge is, these CAR T cells have been successful in liquid tumors, mainly because the liquid tumors are easily accessible and, the targets are well defined. And for solid tumors, it's been a challenge. But, right now we see a lot many trials. The technology has changed in 2022. I think with better technology and better screening of patients, better screening of targets, I think there is a way that they can develop CAR T cells towards this. And I fervently hope that we can make immunotherapy work in medullary thyroid cancer, because targeted therapy works, but it works for some time. Immunotherapy does not seem to work, but if it works for a long, long time. So how can we, synergize, right? Synergize both these combinations, either immune checkpoint therapy or CAR T cell therapy. So my vision is this. So probably if a patient comes in with a bulky thyroid cancer, I think you definitely need targeted therapies to debulk the disease so that you can get rid of the disease quickly. You can debulk the disease and lessen the disease quickly. Beyond that, once you go on a maintenance phase, probably you need the immune system to kick in to get rid of the rest of the cells. You can't, again, ideally 10, 20 years from now, you don't want to be on a treatment forever. Right. So I think that's when immunotherapy kicks in. That's when these technologies like CAR T cell therapies kick in. And I fervently hope that they work. Right. I think it's great they work in mouse models. We haven't seen that in the clinic yet with patients. They're just starting. So again, we keep our fingers crossed to see how we translate that into human trials.
Yes, that's very exciting. Ben wants to tell you thank you very much for answering his question. And I did forward your email to him. Do you have access to that screen? Is that still open that had your contact info on it, Dr. Subbiah, in case somebody else wants to?
Yeah, I'll just post it again. Let me pull it up. This one? Yep.
That one.
Yes.
While everybody's jotting that down to contact you, how did you get into targeted therapy and research? When did you decide that's what your path was going to be?
Again, as a trainee in oncology, I trained at MD Anderson, and I've had people who inspired me. Dr. Emil Freirich, the late Dr. Freirich. He was 92 or 93 when he passed away like 2 years ago. Dr. Norman Jaffe, who passed away last week. And of course, Dr. Russell Kurzrock and many others, right? Again, when these senior leaders, they brought in, the drugs like chemotherapy and all this to clinical trials in humans. And, what I found during my training was, giving the same drug and over again and trying to find different answers is not going to work, right? We need to do new things. We need to do different things. We need to continuously challenge what is existing, right? I think that's been my motto, and that's a thing that has been tattooed in my memories. I need to personally keep challenging myself, personally keep challenging the customs and the norms, and always be humble and modest in terms, because cancer is like a giant beast, right? We need to work together for this. I think that's several people. There are so many people to be thankful for the inspiration they've given me. But what I wanted to do is always look for something different, look for new things, so that we can move the cancer needle forward. I think even if each and every one of us moves at least 1% in our lifetimes, it adds up.
That's awesome. Oh no, you've got a message here from Todd, and he says, Texas Todd says, you're the best doctor on planet Earth, and thanks for keeping me alive.
All right, Texas Todd, I'm going to take a picture of this and show it to my wife who's standing beside me. Thank you so much.
Wait, show it to your wife because why? Because
No.
you're not her favorite? No. For those that don't know, I'm pretty sure Dr. Subbiah's wife is also
Oh,
an
she's
MD
fantastic.
at
Yeah, she's an MD at
MD.
MD Anderson. She's fantastic. She works in the palliative, rehabilitative, integrative medicine, and she works with this thing called patient-reported outcomes. Right? She's more engaged with patient-reported outcomes, and that's one of my favorite areas to include patients in design and conduct of clinical trials. See, that's not happening often. Right? When we design clinical trials, we don't include patients, and I really want to include patient input on that.
Right. Are there more questions out there, anybody?
I think it's always good to give time back on a Saturday afternoon, right? I'll be happy to answer any questions later.
What are your thoughts on a 3/4 or half dose?
I think that's a great question, right? So the main thing is that each one of us is different, right? And each one of us, the PK is different. I think the FDA has also said that we need to customize the doses for patients. And some patients cannot tolerate the 160, whereas some patients can easily tolerate that. I think anything above 80 or 120 would be reasonable. I think we can definitely customize the dose for patients. Absolutely.
Do you think they'll, without having a grade 3 adverse effect, do you think the trial would, if those of us that are still on trial, they would say, sure, let's reduce your dose?
Yeah, no, absolutely. I think that's a good question. I think we need to work with you and Texas start for these questions, right? Because again, as a trialist, I'm not taking the drug, right? As a doctor, I'm not taking the drug. You are taking the drug. You feel that every day, right? With we are used to old generation when I trained, right? We are used to giving chemotherapy. We give one dose, all hair falls out, and then the counts go down, and within 4 weeks we do the same thing, right? They recover and we do the same thing. But these are drugs that are given chronically, right? And you're taking it every day. So, if you have any prohibitive side effects, it's okay to hold treatment. It's okay to dose reduce. We can work you can work with your physician to, customize the dosing for you. Because, take for instance, simple Tylenol, right? For me, I need to take, 2 full doses of Tylenol for me for my headache to go away, whereas my mom can take just 1/2 a pill and her headache goes away. Right? So these are individual customized even for simple Tylenol. This is a complex cancer, right? So I think we need to, again, although the FDA approved dose is that, we should definitely customize the dosing for patients. Absolutely.
I like it. I've got another
and the clear thing is that's why we need your input, right? And clear input into these trial designs, not just during the phase, but beyond like 1 or 2 years. So we don't know what the chronic side effects are going to be beyond 1, 2, 3 years of these drugs, right? These are new drugs. Yeah.
I've got another question for you. Do you think the latest changes to limit what can be charged for medicine will affect trials in the future?
That's a great question. So we don't know the implications of that, right? I think those are going to be only for specific drugs, as far as I know. And they still have, the good news is that the government and the FDA has incentives for rare diseases, orphan diseases, right? Which have been the stepchild, like pediatric cancers as well, right? What happened was 20 years ago, 25 years ago, companies used to develop drugs only for lung cancer, breast cancer, and common cancers, because cancers like thyroid cancer or cancers that I see these are rare cancers. There was no incentive for them to develop drugs. So the good news is that the government has time and again have so many programs, right? Orphan disease status, breakthrough designation, and all that to incentivize drug development. I'm sure those will ensure that all the pharma companies that that have the major resources will continue to develop drugs in these rare diseases. Yeah, that's good. Now we got to step up for the stepchild, and again, there are definitely laws that help with rare diseases.
I don't know if you really want to take another question from Texas, Todd. but here it goes. Some say max dose can accelerate progression, so a reduced dose might cause longer success. What are your thoughts on that theory or debate?
So the key is max dose. See, thing is that we need to hit the receptor hard, because sometimes if we go up on a lower dose, the cancer develops resistance, then the game is already lost. So I think we need to come up with not the maximum dose, but not the lowest dose, but an optimal dose for every patient.
We really appreciate you. We've been wanting you to speak for several years. And now that we've got you, you can just put that in your calendar, because we're going to want you to come teach us more every year. Okay.
Oh, thank you so much. I'm looking forward to working with all of you when we design the next generation studies. Thank you.
Thank you very much.
Thank you so much.
Thank you very much, Dr.
Thank you.
Subbiah. Have a good weekend.
Thank you.
Thank you, Dr. Subbiah.
Thank you so much, Gary. Thank you. Thank you, everyone. Have a nice weekend. Bye-bye.
Take care.
Thank you, everybody.